Hello, everybody. I'm from the FDA. I've come to audit you. No, seriously. I am laboratory manager here at the BTCCRC. In our laboratory, in the bioanalysis laboratory, we develop, validate and apply chromatographic methods to PK studies that we run here at the Royal Brisbane Hospital, across Brisbane, Queensland, across Australia and across the world. And our interest in microsampling can probably be traced back to one of the multi-centre, multinational studies where there was, picture if you will, plasma samples strewed across freezers across the world and a whole heap of dry ice, a whole heap of shipping, very complex procedure getting everything back. And I thought there's got to be an easier way than shipping everything on dry ice. Now, so we're thinking about microsamples. But previously I was laboratory manager at a local CRO, participating in the bioanalysis of about 100 pharmacokinetic trials, generic drug trials for bioequivalence. Most of those studies went to the TGA for approval. Some of them went to the EMEA, some went to the FDA. And part of my role was responding to the regulatory concerns about the bioanalysis. So I'm talking to you today about the regulatory aspects of microsampling. Now, first up, I'm going to stress that even if you're not trying to develop your own drug and get it marketed, there are aspects that I think would be of interest to you. So even if that is what you understand a regulator to be, it's meant to be in the mouth, by the way, there is something for you here. I'll be talking mainly about the FDA as the regulator. Then we'll be talking about what you could do about it, and then where it's all going. So, if you are seeking approval for your drug that requires bioanalysis, you'll probably be familiar with the FDA guidance for industry on bioanalytical method validation. And that contains in it a number of various rules or guidances. A central one is the what we call PANDA in our lab, which is precision and accuracy. The intra and inter-batch PANDA, and the test that they have in there is, to measure 5 replicates at 3 concentrations, and the criterion that's in the document is that the precision and accuracy have to be within 15%. So using liquid, normal, traditional sampling, that's what everyone's familiar with. If you are wanting to move to microsamples, what more do you have to consider? Now, if you aren't developing a drug, hands up if you're not developing a drug, but you might want to publish your work, okay? The peer review will expect quality results, and the peer review generally wants bioanalysis in accordance with the best practice. Even if you're not publishing, you don't want to generate misleading results. You want to avoid avoidable errors and follow best practice. So, this takes us back to the guidance. And with these guidances, I'd say that the FDA wants to encourage people to use new technologies, and they have to be done consistently with the goals of the regulator. So the FDA can take the credit for the goal of safe and effective medicines, and possibly the blame for some of the torturous sentences that are in the document. But the content is a distillation of the best practice that often comes from the practitioners in the field that the regulators observe. And so the regulatory guidance can be a mirror that reflects the best practice. And this can be useful. So the guidances can be useful for all practitioners. specifically for microsamples. The first time I've seen it the earliest in a regulatory document is the 2013 FDA draft guidance. And they mention dried blood spots, and they highlight specific issues like storage and handling temperature, which we've discussed already today. The homogeneity of the sample spot that we've mentioned already. Hematocrit stability, carryover, we've all mentioned that. And then the incurred sample reanalysis. And that's, speaking from a laboratory basis, that's a really interesting test to follow, because when you're working in the laboratory and setting up your assays, you're normally working with a spiked sample where you've got some blank plasma and you put your drug in, whereas an incurred sample is one that you take from a person who's had the drug administered to them. So it contains things in there like metabolites that we often don't see in validation unless we specifically put them in there. Now those tests I've mentioned can usually be done in the laboratory without any further messing around. But they also mention correlative studies, or the bridging studies, that are particularly problematic for laboratory folk, because it's not a bench test, and this often requires parallel sampling, like having the microsample and a traditional sample taken at the same time and then comparing them. Now, unfortunately for this document, unlike the one we're very familiar with, it does not provide any description of the tests for microsamples and any description of the criteria for microsamples. Although, now there is a more recent document by the ICH, which is the latest word from the regulator that I can find. And even though it's issued by the ICH, it does specify it's the thinking of the Food and Drug Administration. Interestingly, this is a note for guidance on toxicokinetics. And toxicokinetics, this is driven by the massive benefit for preclinical animal work that microsamples promise. Because there's a limited volume of blood in animals when doing this work, and by scaling down the sampling, then there's massive benefits to be obtained. Pardon me. So the pressure to implement microsampling in the regulatory space is coming from the toxicokinetics in the drug development pipeline. Now, as Chris mentioned earlier, the 3 R's of replacement, refinement and reduction. What we found in our laboratory over the years talking about microsampling is to talk about the 3 S's. And when I talk about the ICH recommendations, there's the 3 S's there. That's the system, and that would be the animal, or in our case, the patient being studied. There's the sample, and that's how the sample is removed from the system, and that needs to be representative. Oh, thank you. The sample needs to be representative of the system that you're taking it from. And in our parallel microsampling study that Susie mentioned before, we take a central venous sample or arterial sample, and then there's the specimen. Now, the specimen is the format of the sample that is measured, and that needs to remain viable. So in our parallel sampling study, we take 1 sample, but we generate 3 specimens: the liquid plasma, the plasma VAMs, and the whole blood VAMs. I've coined that now, so if you want to tweet me to fame, hashtag 3S is awesome. So the liquid specimen We've talked about most of this already. The liquid specimen. They specifically mention sample homogeneity. Like if in your capillary, do you have the same sample at the top or at the bottom? Issues with small volumes when you freeze it. As we've talked about before, do you have enough sensitivity? Do you have enough femtograms of sample? And then also, is there an effect of dilution from the anticoagulant? For the dried specimen, there's our familiar hematocrit uniformity and incurred sample reanalysis. But they do acknowledge that VAMS type sampling, where you have a fixed volume, controlled, can help address some of the issues. Regarding the sampling process, they still say that bridging studies are important, and you have to compare your microsample against your traditional sample. Although they do say that this bridging study can be omitted in certain circumstances. For example, when you have the same assay conditions, the same matrix from the same site. So if you're doing an animal study and you've been taking whole blood dried blood spots from a tail, going to whole blood VAMS from a tail wouldn't require well, you could justify not doing a bridging study in that case. And finally, they do acknowledge some of the various influences that can affect your sample, such as plasma binding and things like that we've already talked about. And finally, they mention the system, and it's kind of like a physiological equivalent of the Heisenberg uncertainty principle, where the act of observing a system changes that system. So that, is the sampling responsible for any of the effects? And in some way, this is an argument against traditional sampling, an argument for microsampling. If you have a very small animal and you're pulling lots and lots of blood out of there, you're better off. And that can have effects for the animal. So microsampling can be of great benefit. So, where does that actually lead us? There are no defined tests in that question and answer document. They don't provide any acceptance criteria. In effect, the FDA has told us that the cat has to be skinned. Now, it's up to us. We have to work out how many and what type of cat. And we have to defend how well we have skinned it. So where do we find our answers to skinning a cat? I think the answer would be to look at the practitioners in the industry and in academia. There's a lot of information. It's growing every year in the literature. My particular favourite, I'll give a shout out now to the European Bioanalysis Forum. For many years now, they've been producing recommendations and papers. There's also focus sites where, like Bioanalysis Zone, I like that one. There's some 3R websites that have a lot of useful information. And I also think we shouldn't discount the expertise in this room and meetings like this. So, if I had to make any recommendation about the tests that are not in the guidance, that the regulator's not telling us about, the features I think would be that it obviously has to be relevant to the application which you're using. You'd have to find it to be meaningful to what you're doing. You'd go insane if it wasn't meaningful. And try to make it routine so you can have it in your workflow. and it won't hurt that much. And finally, if it's defendable by pointing to some prior art, like a paper that is well regarded, or another test that might appear similar in the guidances. As a generic kind of test, you could, for a specimen type test, you could think of the test condition, if you're looking at spot volume, your central tendency, like maybe the volume that you're using for your standards, and then extreme volumes either side. Look at two analyte concentrations, a low one and a high one, 3 replicates, and have results within 15%. There's enough stuff like that in their guidance that you'll get away with that. Bridging studies, I think, are a more complex answer that, at least when you're dealing with patients, is a lot more effort. I might just to Christophe Stover from Ghent in Belgium, who's early this year published this paper where he pretty much does everything, looking at a linear regression. And we've talked a little bit about the agreement between a dried blood spot. By the way, he used dried blood spots and central plasma. It's not exactly a 1-to-1 ratio, it's a 0.85. He also looked at a passing Bablock scatter plot, which is the one in the diagram. He's done a Bland-Altman analysis and a Pearson correlation coefficient. He's followed that up with the incurred sample reanalysis, and also found a good correlation between capillary and venous concentrations. So the future, I think there's 2 things highlighted there. Adequate validation is one of the things to get you through, but really I think the process will be driven mainly by the practitioners, and I think the toxicokinetic practitioners have more at stake and will push harder, and they'll convince the regulator of the right way to do this and what type of tests are better defined to get us there. And then ultimately, starting at the start of the drug pipeline with microsampling, it will become accepted practice and no longer novel in the regulatory pipeline. And so until then, I think we just keep working and keep coming to seminars and go scuba diving. So, thank you.